Today we are very pleased to welcome Dr. Cheryl Lichti, Assistant Professor in the Development of Pathology and Immunology, as well as the Head of Mass Spectrometry Laboratory and Manager of the Bursky Center for Human Immunology and Immunotherapy at Washington University in St. Louis. Cheryl obtained her Ph.D. from Duke University in organic chemistry. In her career, Cheryl has held positions at multiple institutions, but most recently, prior to joining Washington University in St. Louis, Cheryl was at UTMB as a fellow in the Mitchell Center for Neurodegenerative Diseases. Here she focused on neuropeptidomics and neurodegenerative diseases using quantitative proteomics, analysis of PTM and single amino acid variants. Her current research focus is in immunopeptidomics, specifically on type 1 diabetes and cancer immunology. With over 10 years of experience in method development for both small molecules and peptides, Cheryl has widespread experience working in multidisciplinary teams to design mass spec experiments for solving research problems in multiple biomedical disciplines. So Cheryl, I'll turn it over to you and you can begin sharing your screen.
Thank you, Jenny. I'm working on getting it up now. Here we go. Oh, I cannot start while you're sharing. There we go. Well, maybe. Let me see here. I think this is what I want. Okay, so if I did this right, you should be seeing my slides now. Or did I do it wrong?
No, we can see them. It's all good.
Okay, cool. So this is my title slide, of course. So obviously I'm going to be telling you about the immunopeptidome. What I'm going to tell you about first, though, I'm going to start with a brief overview of type 1 diabetes, and then I will talk about the work in our recent publication from Nature Immunology that just came out earlier this year. So type 1 diabetes, as most of you probably know, is an autoimmune disease, and it's triggered by destruction of pancreatic beta cells, which produce insulin. And the disease is initiated by presentation of insulin-derived peptide fragments. And there is a strong genetic component to this disease in humans. Those who express the allelic variant of MHC-II, that is HLA-DQ8, are most likely to get the disease. And there is a mouse model, the non-obese diabetic mouse model, that expresses an allele that is very, very similar to HLA-DQ8. It is IAG7. So when I talk about IAG7, you will see that the peptide presentation is very similar. So from earlier work that was done in the Unanue lab, which is the group I'm working with on this project, what is known is that in pancreatic beta cells, upon stimulation by glucose, insulin is released. And this insulin is mostly released in the form of dense core granules, which are mostly comprised of insulin, but have other proteins in as well. But as a natural part of homeostasis, some of these granules are degraded by fusing with lysosomes in the process of crenophagy. They produce these bodies called chromosomes, which they are made up of degraded insulin peptides. And so some of these are released. And in our previous study that was published in Nature, we showed that these peptides that are produced in chromosomes actually are released into circulation and they are seen in the periphery. So therefore they are involved in the onset of type 1 diabetes. So the question that we have for this current work is, can we actually identify some of these peptides in their MHC-II bound form by mass spectrometry from actual biological samples? And so we proceeded to take on this project. So the person shown at the top here, Tony Vomand, was responsible for isolating the peptides that we looked at from islets, from spleens, and also from pancreatic lymph nodes in the NOD mouse. So I am comparing these results from earlier work that we had, which we looked at chromosomes and also the peptidomes generated by exocytosis. I then analyzed all of these peptide mixtures by mass spec, and then Tony then did biological studies on synthetic forms of these peptides to test binding to AG7 and in some cases to identify the registers. And then we also looked for CD4 T cells to these same peptides, and also looked for diabetogenicity of these peptides. So to start with, to look at an overview of the peptides that we looked at. So for any of you who are familiar with immunopeptidomics, for class 1 peptides, you mostly identify smaller 8-mers and 9-mers. In the case of MHC-II, the length of the peptides identified is longer, 14 mers to 17 mers typically, and as you see, the peptides that we identified follow this trend. The other important feature that is seen for what we identify is what is shown in the Gibbs cluster analysis. What is known in earlier work from the Inanaway lab is that the P9 position of the peptide shows a very, very strong preference for an acidic residue. At P4, there's also a preference for a hydrophobic amino acid residues, side chain, notably isoleucine, leucine, and valine. And so you see that our peptides in the sun-supervised analysis demonstrated a match with the known binding preference for these peptides. Now, in terms of beta cell protein, or more specifically peptides that we identified, this table demonstrates the families, what we identified. So to back up, remember I said that the peptide sequences vary and link somewhat, but they tend to cluster in what we call families with peptides of varying lengths representing the same portion of the sequence. And so what you see on this table is a comparison of what we found in the MHC-II bound peptidome, comparing to what we identified in the free peptidome, and this was in our previous Nature paper. We looked at dense core granules, chromosomes, and also the secreted peptidome, and this comes from cultured islets, challenged with insulin, and then analyzing the supernatant to see what peptides are secreted. And the quantification here in terms of relative abundance comes from summing the abundances actually that we got from peaks for each peptide in the family, and then dividing, we scaled relative to insulin 2C peptide. So in this case, that would be the abundance, sum of the abundances of the 16 peptides identified in islets. Here, that's 38. So in all cases, relative to insulin 2C peptide. This is relative abundance in the islets. So as you would expect, insulin 2 is the most abundant thing. Other proteins from the islets that are not as abundant are seen in lower abundance, relative abundance. Now, looking at these results in a different way, we're looking more at the immunology side of things. And so what we noticed, interestingly, is that if you look at the peptides that are identified in both the islets and the periphery, aka the PLN and the spleen, versus the islets only, what you see is that overall, for most of these, we actually see greater T cell autoreactivity for those that are also identified in the periphery. That is evidenced by greater production of IL-2 and interferon gamma as measured in an ELISpot assay. You also see slightly better binding and better overall abundance for most of these. Now, the other thing that is really interesting, and the first thing we found here, so bit of a backstory. Initially, when we did these searches, we did not look for deamidation, but then I started noticing when I looked in Peak's PTM searches that there were some very interesting deamidations. Sorry, my office light likes to go out and I have to wave my arms. So what we saw specifically is some deamidation in the C terminus of insulin 1 C peptide that was not seen in insulin 2. And the only amino acid that is different here is an arginine in INS 1 versus a glutamine in INS 2. So there are lots of reports in the literature of deamidation being important in type 1 diabetes, in some reports for autoantigens with deamidation, but more of those are toward the N-terminal side, as we see here. There are no reports C-terminal. And what we believe here, there are also reports of this being a biological process with tissue transglutaminase. We believe this is spontaneous because from really early studies that were done, the sequences around these peptides that are deamidated don't match the known sequence preference for transglutaminase. And especially on the C-terminus, it's not, there are residues that are on the C-terminal side that are important for the enzymatic process. And obviously C-terminally, those are not there. And so this has to be a spontaneous project. The other sidebar here is that we actually, to make sure that this was not an artifact, put in the C-terminal peptide that's highlighted in brown, put it through our entire purification process, and we didn't see deamidation. Therefore, we know this is occurring biologically. And we were especially intrigued by this C-terminal deamidation because if you compare the sequence of this peptide with what we saw as and what we know about binding preference for the DQ for AG7, you see that deamidation should make this peptide bind a lot more strongly. We also have the requisite leucine at P4, and we have a valine at P6, which is also sort of preferred here. So we followed up on this. Now, the other thing, I'm showing you a representative sequence that we got from Peaks. And what you should notice here is that from the information we have here, we actually don't have enough information to determine whether the modification is here on the glutamine or on the arginine that precedes it. For us, that is important because Citral A Nation is also a possibility. Citral A Nation has the exact same delta mass associated with it as deamidation, and in previous work from the Unanaway lab, it has been shown that there is some biological activity associated with Citral A Nation. In fact, it would be in a T cell contact residue rather than in the binding residue if it was citrullonated. So we actually got synthetic forms of the citrullinated versus the deamidated peptide, and you can see the two spectra side by side here. The other thing that is obvious is that the spectrum for the deamidated synthetic standard is a much, much better match for what we observed rather than citrullination. So we have confirmed that we are actually seeing deamidation. Now, the other interesting thing to come out of our work has to do with hybrid insulin peptides. Now, hybrid insulin peptides are by no means a new thing from our lab. They were reported from the lab of Catherine Haskins in Denver. She has several papers talking about these. And the way they identified these hybrid insulin peptides is that they took islets and they digested them with Glu-C. So they did a proteolytic digest and then analyzed by LC-MS and tested for T cell reactivity in order to identify HIPs. So there is no mass spec-based identification of hybrid insulin peptides in these studies. In other words, no identification of the bound form of the peptide. But they did identify a couple of important ones, specifically the one on the bottom, which is highlighted in red, this insulin 2C chromogranin A peptide is actually an antigen for a very highly diabetogenic T cell line, BDC2.5. So anytime we've published any of our work with the peptidomes, we've been asked questions about whether or not we have identified any of these hybrid insulin peptides due to the work of Haskins' lab. And very luckily for us, as we were analyzing these samples, the Haskins lab published an in silico database that they generated for hybrid insulin peptides, in which they computationally took various truncated forms of insulin C peptides and coupled those with various natural cleavage products of various abundant beta cell proteins, insulin, chromogranin A, islet amyloid polypeptide, and neuropeptide Y specifically. This database has over 3,000 entries. And so we made use of this database in our work. So the approach that we used was to actually do a multi-round search. So we did an initial search against the mouse proteome, and then filtered at 1% false discovery rate at the peptide spectrum match level. Because when we're looking at immunopeptides, we really don't care about protein IDs. We care about confident peptide IDs. So I always filter at the peptide spectrum match level and pretty much turn off a requirement for a protein score, but require a significant peptide level score. The other thing I tend to do is that, so when I'm looking at an entire MHC peptidome, I look at the 1% false discovery rate. But when we're looking specifically for beta cell ones, especially insulin derived, I will often drop the false discovery rate down to 5% false discovery rate and manually verify, because there are known issues with false discovery rate for peptidomics. A lot of times things that are real and interesting will fall below the FDR cutoff. So in any event, I do this search, go through a multi-round, and then look to see what I identified. Now, if you looked closely at the table I showed you earlier, I did have this insulin C islet amyloid polypeptide HIP that we did identify, and we verified it using a synthetic standard. This was identified in islets and in pancreatic lymph nodes. The representative spectrum is shown here. You can see that's a very nice match. Now, interestingly, when we did this search initially between islets and pancreatic lymph nodes, we actually had 5 hybrid insulin peptides that were tentatively identified. However, when we looked at the synthetic standards, not all of them matched. In fact, most of them were false positives. So you can see here, this is a terrible match, right? So the top spectrum is the one that was tentatively assigned initially as a hybrid insulin peptide. On the bottom, you see the spectrum for the synthetic standard. It's a terrible match. And what you see here is, if you look closely, that is an oxidized methionine. And there are many, many peaks representing neutral loss of SOCH4, which you would expect from a peptide containing an oxidized methionine. So this was a terrible match. So being a naturally curious person and wanting to know why the heck the spectrum was so misassigned, I traced back to the original scan number for the spectrum that was assigned to the hybrid insulin peptides. And I went into the peaks, into the spider search that was basically the result of the Ph.D search, but that I did the multi-round from, to see if there were any alternate assignments for this spectrum. And it turns out that there were. It turns out that I found a sodium adduct to this very, very abundant C-peptide fragment. And it turns out that the unmodified form of this peptide, in other words, just protonated rather than sodiated form of this peptide, was present. Exact same retention time as the sodium adduct, screamingly abundant. And if you look at the spectrum, this actually makes a lot more sense than the earlier assignment, because if you go back and look, note that there's a very large stretch of amino acids toward the C-terminal end where you don't have fragment ions. For this one, you do have them. And moreover, if you look at the other HIPs the other four for which we did not have matching synthetic standards, we saw the exact same thing. We typically saw either dehydration or sodium adducts of very abundant insulin C-peptide fragments. And again, in all cases, the retention time for the modified peptide matched the unmodified form. So dehydration is something that you might expect to see happening in source. If it is happening that way, you would see coelution, and that's exactly what we saw. So what ended up being our workflow for identifying HIPs, and is what we used going forward, was something that looked like this, where we took our data files and did a peaks search. From the original peaks search, we went forward with a multi-round search against the HIP database, and we exported the list of peaks DB searches. So we got every possible peptide spectrum match. And from this, we pulled out those peptide spectrum matches for hybrid insulin peptides. Also, we took the spider search result that came from the same PeakCB search, got the list of database search peptide spectrum matches, and then collated these by scan number in Excel using the VLOOKUP function. So basically, for every single peptide spectrum match we had for a hybrid insulin peptide, we looked here in an automated fashion to see if we had anything that was likely a modified insulin peptide. From that, we got the final list of HIPs for further verification. So it filtered down the list significantly, and the ones that were found in this manner were verified by synthetic standards. So in conclusion, what I can say is that using peaks, we have always been able to find important PTMs in the MHC-II Peptidome of the NOD mouse. So I didn't mention it specifically in this talk, but for insulin B, 9 to 23, which is a very important autoantigen. It has a cysteine, and we couldn't find it, couldn't figure out why until I looked in Peaks PTM and discovered that we were seeing it in multiple forms depending on where the samples came from. It was either an oxidation to cysteic acid, which is what we look for now, or you can also see it as a glutathione adduct or a cysteine adduct. And so in addition to that, in looking for hybrid insulin peptides, which are very important and hot these days, we have found that the Peaks, PTM, and Spider searches give us valuable information regarding spectra that need to be filtered out because they're more likely to be false positives in our hit searches. At this point, I would like to acknowledge the people who did the work on this project, the key person here being Dr. Emil Unanaway, who is the PI on the grant. Sorry, office light shut off again. Xiaoxiao Wan, a research scientist in the lab, has done a lot of the work, and Anthony Vomand and Orion Peterson are responsible for the technical work that you saw in terms of all of the oscillations. Other lab members have contributed valuable discussion. For financial support, they're listed there, and I would like to thank all of you for your attention and answer any questions you might have.